Alright, so we're going to work today on the same chapter, right? We're still on lipids, have some worksheets, and we're going to look at lipids from a slightly different perspective today. So some of what you're going to see is going to be the same information as the other lecture, and some of it will be different. What's the same will be from a slightly different angle. So we're going to look a little more at biological function today and have a little bit more graphics. So if we're looking at lipids, and we had listed some functions the other day of our lipids, again, we're looking here at a plant and the wax coating on the outside of the plant. Also important is giving us energy, so energy-rich compounds that have very low density. So that means that, a lot less mass, right? Mass per unit volume. And we need to store energy. And then we already know cellular membrane components. So this is a different way at looking at classifying our lipids. We can look at the broad category of our lipids, and then we can separate them out in the tree into ones that are saponifiable and ones that are non-saponifiable. And so we've talked already that our saponifiable lipids are basically a hydrolysis reaction, and we're using NaOH or KOH, we're using a base. So that chemical reaction can actually split out how we're categorizing. Our non-saponifiable lipids then are going to be our steroids and our prostaglandins, and our saponifiable lipids break down into simple and complex. So here, of course, we're just looking again at the structure, how many parts and pieces are attached, and therefore break it into not so many parts and pieces and a lot more parts and pieces category. So our waxes and our triglycerides are going to be in our simple category. Our phosphoglycerides and our sphingolipids are going to show up in our complex category. So now focusing on our saponifiable lipids, we've talked about how we're in base solution, or NaOH, KOH, hydrolyzing them, so breaking apart that ester linkage to go back into our carboxylic acid and whatever else we're producing. So our simple lipids, our triglycerides and waxes, are saponifiable. Our complex lipids, and we had said our steroids and prostaglandins, are not saponifiable because of their very differing structure. They don't really have that ester linkage in them. So we look at a lot of ester hydrolysis and then a few where we don't really talk about ester hydrolysis. So our fatty acids that we talked about show up, as we saw the other day, in a lot of our lipids, and we know they're long-chain carboxylic acids with those very long nonpolar tails that we looked a little bit at. And when we think of fats and oils, when we think of oil and water not mixing, we're really thinking of those long nonpolar tails that are creating that atmosphere, creating that chemical structure that we think of as that oily layer with the nonpolar. And we know we have the polar head that we've talked about that's coming from our carboxylic acid group, and we draw them in a model that's very simple and easy to draw. Down here we can see this. We just take and draw our nonpolar chain as basically a squiggly line, and then, because it's a tail, right? And it's a squiggly line because if you think back on our saturated fatty acids, tetrahedral, and we said we basically draw the chain as going up and down and up and down, and we'll look at that in a minute, kind of that folding of the paper. Our polar head, we think of our heads as being essentially spherical at some level. So we tend to just draw the carboxylic acid group, although it's obviously not spherical as a spherical type group. So that we know that our straight chain, no double bonds, actually has this format, and so that's where that nonpolar tail idea comes from. Alright, so I go back to the clicker so I can advance the slide. I love this picture. This is what we talk about both with how does soap and detergent tend to work, as well as what's going on in our bodies. And so we talk about micelles, and so this spherical structure is called a micelle. We can see that all of the nonpolar tails are pointed in towards each other. We know like dissolves like, so we know like mixes with like, back to Chemie 121. So we know that those nonpolar tails are all going to want to gather together. And if we're in a solution of water, the nonpolar tails will gather together and the polar heads are going to be sticking out because they're polar, they're going to go with the water. If you were to take these same chemicals and put them in a fatty solution, they would of course do the opposite and stick the polar heads to the inside to protect each other, and then the outside is that sphere would be the nonpolar tails. Most of what we look at in our environments are where we really want the polar heads pointing out and the nonpolar tails pointing in. So we know if we're polar, we're hydrophilic, we're water-loving, and if we're nonpolar, we're hydrophobic, phobia of water, so we come back to that. And also this works with things like our soaps and detergents, where a speck of dirt is typically nonpolar. The nonpolar heads can group around it, and then the water from the sink or the shower can wash away the sphere because the polar head's connecting with the water. But you can see here a lot of biological functions, including transporting other lipids, since they're more the fatty acids, in the blood in a micelle fashion, because they can group together, and cholesterol transport actually involves that type of design. So we've already looked at fatty acids, so this one is just a little bit of a review that we know our unsaturated fatty acids have those cis double bonds typically, and our saturated fatty acids are not going to have double bonds. They're saturated with hydrogen, so all single bonds. Typically we're talking about a straight chain, no branches coming off, so just one horizontal line left to right makes it easy to count our carbons. And again, we can look at what some of these fatty acids look like. So again, this picture we've seen with the kinking of the chain when we get the cis carbon-carbon double bond, because it pulls everything to the same side. And the more cis double bonds, the more kinking we're going to have, and we have seen how that affects melting point. They can't stack as tightly with the kinks in them, and therefore they're going to melt at a much lower temperature. And so that kinking actually we know is related to our vegetable oils versus our animal fats. And the more double bonds, cis double bonds, the more kinking, it's going to melt at a lower temperature, and it's going to be an oil. So again, we're just seeing a long melting point chart and which kind of oils it comes from. So this is similar to the chart we saw in the last PowerPoint. It's just organized a little bit differently. Instead of the source being what's important, we'll notice this is grouped more by the name, and then the source is kind of off to the side along with the melting points. So we talked about essential fatty acids and linoleic and linolenic acid being essential. We can get them from plant and fish oils, and so we hear a lot about taking fish oil to get our essential fatty acids. We talked about omega-3 and omega-6, looking at some of what they actually do in the body. Hormone-like substance, so we're regulating our blood pressure, our blood clotting, blood lipid levels, immune response, inflammation response. So these fatty acids are really, really important for our body, so it's pretty critical that we're getting enough of them somehow. There are other omega-3 fatty acids and other omega-6 fatty acids. We talked about the nomenclature, and so as long as you have a double bond, 3 in the first double bond, 3 in from the end or 6 in from the end, technically it's an omega-3 or an omega-6 fatty acid, so it can be a matter of which omega-3 and omega-6 fatty acid. So if these are essential, we know we don't produce them in the body, but we can use these to produce other ones that we need in the body. So we know we've looked at our fats being our solids, and our oils tending to be from more plant and fish sources, tending to be more of the monounsaturated and polyunsaturated fats. So this is just a really nice general picture of like, how do I recognize I have fats and oils? How do I recognize I have a triglyceride or a triacylglycerol? You'll notice all of these names are used interchangeably, and you heard me stumble a little on that the other day in the other PowerPoint. But you can see here that we just have a molecule of glycerol, ends in OL, right? So we know it's an alcohol. And so whatever glycerol is, we know we have it. In order to connect our fatty acids, it actually has to have 3 OH groups on it at this juncture. But we have a molecule of glycerol, and to it we can hook 3 fatty acids. So just the idea, we can take a very simple diagram and use that to try and help us remember triglycerides, tri for 3, so 3 fatty acids hooked to glycerol. Triacylglycerol may be a little easier to remember with that name, since the word glycerol is just directly in there with the tri. So you'll notice the first bullet point here where we say rarely identical. So basically that means that when I take that glycerol molecule and I attach my 3 fatty acids, usually all 3 of those fatty acids are not the same molecule. I could take stearic acid and glycerol, and I could take 3 stearic acids and hook all 3 of those to the glycerol, so I'd have glycerol and 3 identical fatty acids attached. But typically all 3 are not the same, maybe 2 are the same, maybe all 3 are different. So you can see in the picture down here, that is exactly what we're looking at, where we have a saturated version right here, so no double bonds. Here I have 1 double bond. Here you'll notice this little 2, so that means I have CH2, CH double bond CH, CH2, CH double bond CH, and then it continues. So using that short form nomenclature we talked about to save on writing, so this actually has 2 double bonds in it. So obviously different molecules. We know we can undergo hydrolysis and split them out. We've looked at that, or saponification with base, where we're going to break it here, adding our base. We need 3 NaOHs since we're taking all 3 off, and we'll get our glycerol molecule back, and 3 completely different sodium salts, of our carboxylate salts. So here we're just going the other direction, right? So we know we can do hydration, we can do hydrolysis, and break it apart. We can do dehydration and put them together and get our water molecule out of it. So we can hydrate, we can dehydrate, and so we can see in this case we're actually going to, whoops, we'll go back. We are going to go ahead and see if we can use our highlighter correctly. But we're going to go ahead and pull the water molecules out of here and make this attachment right here. So making our ester. So we've seen that ester reaction quite a lot, and that's all we're doing here is just that basic ester reaction. So, structure of fats and oils. Now we're going to talk a little bit about diet. We know we hear a lot these days about how much fat do we have in our diet, is it good or not, what happens with it. A lot of conversations on that, a lot of information coming out, because some people on fairly high fat diets do really well and they're quite healthy, and other people have issues. So we know our carbohydrates, our sugar levels are really connected to that. and the quality of just everything we're eating, but fat definitely plays a role there, as we know. And so we look at it as a risk factor of chronic disease, arteriosclerosis. Blood cholesterol levels is a sign of arteriosclerosis and potentially heart attack. All right, so into the reactions. We know we have hydrolysis. We know we can break these apart, and we looked at saponification on the other page where we break them apart using our NaOH or KOH. Here we're actually using an enzyme. So in this case we have an enzyme lipase. You can see another option is, in this case, this is an acidic solution that we're actually running this hydrolysis reaction. But of course, as soon as we see hydrolysis, we just know, break the ester bond, and we're going to get our alcohol and our carboxylic acid. We're in acid solution, so we know that means we have excess hydrogens in acid solution, and so they're going to bond to the oxygen, so we'll get the carboxylic acid with the alcohol. If we were in base solution doing saponification, if we're in base, we do not have lots of hydrogens floating around, but we do have lots of sodiums floating around with the positive charge, so we get the carboxylate salt instead of the carboxylic acid because the sodiums are floating around in solution. So that's the big thing we want to look for with hydrolysis, if it's in acid or it's in base. If it's in base, NaOH, KOH, then that sodium or potassium ion makes the salt form, and if it's in acid, we'll just get directly the carboxylic acid. So here's our base version, right, that we've already seen, where we can see that the Na plus is going to hook to the oxygen instead of a hydrogen, so we get that sodium salt. We've talked about hard salt and soft soap. Hard salts for hard soap, and then our potassium salts for a soft soap. And traditional soap making's been around quite a long time, right? So traditionally they would just get some animal fat, some lard, they would get some lye, and lye is just non-purified sodium hydroxide, and you can get it out of the ash from a fire. So you just burn wood, right? Just make your bonfire, take the ash, and then put it in water, and then the sodium hydroxide will come on out of there, and you can use that to mix it with your fat and make your soap. But you'll notice here at the bottom, soap making was lost with the fall of the Roman Empire. That seems like a crazy thing. So AD 500 to 1500 are actually considered the soapless centuries, and that's actually related to all of the devastating plagues and the unsanitary conditions in Western Europe because of the lack of cleanliness that was going on then. So it's amazing how something like that could take such a toll on human life and cause such a huge issue. So knowing your chemistry, right, your biochemistry, whether you're in the woods or wherever, it's always good to know some of that, know how to just make some of these basic things, so no matter what, you've got what you need. Hydrogenation, we have looked at. We know often we're using a catalyst such as nickel or platinum, probably the two most popular ones we see, but we know we're just breaking double bonds, alkenes, right? So we're just taking our alkenes and making them alkanes, so same thing here. Obviously, if we're taking an alkene and making it an alkane, it was unsaturated and we're making it more saturated or a heavier fat. And so we can use that to take an oil, like vegetable oil, and turn it into something solid, like margarine, since we know that affects the melting point. We know that makes it harder and waxier. If you go partially, right, you have some double bonds left, but you break some double bonds, you get that smooth, creamy product like peanut butter. So here's the downside, right? While we're doing this process of partial hydrogenation or hydrogenation, our experiment is not going perfectly. And so some of our cis bonds actually change to become trans bonds during the reaction. And it's difficult to control that, and then we get those trans fatty acids. So quality control and the process in order to make the hydrogenation work are really, really critical here. Alright, so triglycerides, this is just our little map. What happens if we add base? We know we make the carboxylate salt with the alcohol, still hydrolysis. What happens if we add acid? Still hydrolysis, breaking that ester bond apart, but we're going to get the carboxylic acid with the alcohol. What happens if we add hydrogen and a catalyst? Now, nothing to do with the ester linkage or the carbonyl group. It's just if there are any carbon-carbon double bonds, any alkene bonds, right? Alkene, we're just going to go ahead and, depending how much hydrogen we have, 2 hydrogens per bond, right? So for every 2 hydrogens, we can change that double bond to a single bond. Okay, back to our waxes that we've looked at in our protective coatings. They do not undergo hydrolysis very easily. It's very difficult to hydrolyze them. So even though they do have that ester bond that we can see, so we know we have, if I click the right thing, we know we have our ester bond here. But we're saying it's not that easy to break it. So it's difficult to break that bond. So these tend to stay together pretty well, which of course, lots of good reasons for that to be important. And so as well as us talking about, oh, you know, the coating on plants, places that we need to use a coating, of course, commercially, we use it pretty heavily in cosmetics, ointments, polishes, anything where we're trying to offer some kind of layer of protection from something. And simple version of waxes, right, to try and help us remember this category. We just got 1 fatty acid chain and 1 alcohol. So our triglycerides had 3 fatty acid chains with an alcohol. This is just 1 of each. Our phosphoglycerides are a form of a phospholipid, so obviously they have phosphate, and glycerol. So glyceride tells us the alcohol, glycerol is in it, that has just the 3 carbon chain with 3 OHs on it. So simple version of this picture where we can see our phosphoglyceride has our glycerol, it's got our phosphate group, it's got 2 fatty acids coming off of it. So we're basically just taking our triglyceride and taking that 3rd fatty acid and replacing it with a phosphate group that has an amino alcohol attached to it. So an alcohol with a nitrogen. So we start out here with glycerol, and we go ahead and say, hey, I got 1 fatty acid, and I got another fatty acid, right? So 2 fatty acids, and then we can add our phosphate group. So we have our phosphate group, we need our amino alcohol, right? So we know right here where we see R double prime, there must be an amino alcohol hooked there somehow. Examples down below, we can see down below, we've looked at these already the other day, but you can see they all have the nitrogen, so we have the amine group. They all have an OH, they have an alcohol group, so they're those amino alcohols. And then just depends which one we hook onto the basic structure we just looked at on the other page. So hook it to the phosphate, and then we have a whole classification of chemicals, biochemicals that do something. So we've talked about our lecithins, they're emulsifying and they form those micelles we looked at. And important in cell membranes. We've talked about our cephalins. They can have either the serine or the ethanolamine, as we saw the other day. Also in cell membranes and very involved in our blood clotting process. So this is just saying, alright, let's look at lecithin. We said that has choline attached. It's our basic structure, and just that R double prime is a choline. And so here we go, here's what it looks like. Cephalin, all we've changed is our amino alcohol at the bottom. Our sphingolipids. Now we're moving into our complex lipid category. And so instead of using glycerol as the alcohol we hook stuff to, we use sphingosine. And sphingosine we looked at already as this big messy molecule down below. So instead of saying one fatty acid and one molecule of glycerol, we now are going to look at sphingosine and then attach other parts and pieces to the sphingosine. So we can see sphingomyelin, and again we have the nice, really simple picture if we look down towards the bottom. So we have, instead of glycerol, we have our sphingosine here, and then we have one fatty acid attached, and then we have our phosphoric acid with choline attached. So again, our phosphate with our choline. So complex lipid because we now are working with sphingosine instead of glycerol, and sphingosine is a very large molecule of glycerol, pretty small and simple. So our sphingolipids that we're looking at, I'm just going to real quick make sure, yeah, I didn't miss anything. Of course, we know that if any of these molecules in our body are not functioning properly, we get some kind of disease. Our body is not at ease, and so if we get too much of something or too little of something, we know that causes us issues too. And so you can see here, what if you have abnormal accumulation? So what if you have too many sphingomyelins, too many glycolipids in the body? What does that mean? And so you can see that you can get Tay-Sachs disease, Niemann-Pick disease. You can see what organs they affect. And all of these mean that your body is accumulating sphingomyelins or glycolipids because the enzyme your body needs to break these down into their parts and pieces and reuse those parts and pieces for other parts of the body is either missing or it's not working right. You can see it's an inherited disease, so we can have mutation, but generally we're looking at a genetically inherited disease here. If we switch to cell membranes, you probably remember from your biology, prokaryotic cells, eukaryotic cells, etc. So we kind of go back and say, okay, what about those? What are we talking about? So we know our prokaryotic cells don't have a nucleus, right? One cell, no nucleus, no membrane enclosed organelle. Our eukaryotic cells, we know, do have that membrane enclosed organelle. And the nucleus certainly would be one of those. So because it's membrane enclosed, that membrane is going to be somehow made out of lipids. And so we're going to tend to look a lot at either lipid bilayers or single lipid layers forming these coverings around parts of our the nucleus and sometimes other parts in the cells to provide that polar-nonpolar separation. So you can see here some cellular organelles. So if we're just looking in like one of our cells, we talk about the endoplasmic reticulum, the nucleus, the mitochondria, the ribosomes. So we're going to look at the fact that all of these need some kind of membrane surrounding it. So we spent a little bit of time on the membrane, and you can see here membranes are pretty heavily lipid, 60% lipid, over half lipid. And then they also have protein, which means we come back to these in our amino acid and protein chapters. You'll notice that we're basically looking at phosphoglycerides, so the presence of that phosphate is very important. Sphingomyelin and cholesterol all play a role. So we know we have good cholesterol, right? We know we need cholesterol in our body. So lipid bilayer is telling us, of course, two layers with the nonpolar tails facing each other. So instead of the micelle, which was that round version, that spherical version with everything pointing to the center, now we're just looking at the two layers, right? So just two horizontal layers, nonpolar tails pointing towards each other, heads pointing away. This implies to us that both sides have water. So when we're protecting something within the cell, the inside of the cell must be watery, the outside of the cell must be watery, but we need to keep the two parts separate. We need things to not just diffuse across or be able to move across. We need separate compartments, and so we're going to have polar parts facing to the inside of the cell and the outside of the cell, and then the nonpolar tails in the middle, and that will help prevent molecules from crossing over that we don't want to cross over. So here's what we call the fluid mosaic model of membrane structure, which you may have seen before. So we can see our lipid bilayer. In this case, it's all yellow. And then we can see we have proteins sticking in there. Those proteins kind of look like eggplant. And some of those proteins, you'll notice, actually have those green chains on the end that have the label carbohydrates. So sugar rings, and you can see they kind of look like the glucose sugar rings we've talked about. Could be galactose sugar rings. Poking out, and we've already talked about how red blood cells have some sugar rings, galactose, on their outside that is part of the process of, is this an A, is this a B? We've of course labeled them depending on exactly what the sugar looks like that's attached. So ultimately we talk about the proteins being embedded because they're transport pathways. That's how we can get things in and out of the cell, and not through the lipid bilayer. So usually we're seeing unsaturated fatty acid chains. We want loose packing, so we want that cis structure with the double bonds, and our membrane needs to be fluid and kind of flow back and forth. If you think about seaweed in the water perhaps, so we need that fluidity in the membrane. It needs some give and not be really hard and rigid. And so if we have that looser, less dense, tighter packing, we solve that problem. So we want those cis bonds to help with the unsaturation. The proteins then are actually kind of floating. In that picture they look stuck, but that word fluid mosaic model, the proteins then are actually kind of floating around in that lipid bilayer, and you can see here the analogy, what about icebergs in the sea? Because they obviously need to be less dense so that they're not just falling through the bottom, but they're actually floating in the solution. Okay, so if we look at our cholesterol ring system, our steroid ring system, this is just such a beautiful, nice, basic version. You can just see clearly we've got those 3 6-membered rings, the 1 5-membered ring all hooked together. Has to be in this pattern. So they're not all horizontal. We can see that we have these two horizontally, but right here we're connecting on the diagonal. So we've got to have that exact structure for anything that falls into our steroid category. Again, that's a lot of carbon and hydrogen, so again, of course, we're looking at nonpolar. And get rid of that writing. Alright, so cholesterol, most abundant steroid, right? We know we make a lot of other steroids from it. We've talked about that. We'll look at some of those pathways. We know it's involved in our bile acids, some derivatives of it, and it also plays a role in our cell membranes. So again, this is just going back briefly over those taking cholesterol and making our bile salts that can in the lipid emulsify lipids in the intestine. So just trying to separate, since we know the lipids are all nonpolar, they're going to want to clump together. We need to be able to break them apart into just small little groupings so the body can help process them. And you can see that that allows the hydrolysis reaction to occur more easily. We know bile is stored, produced by the liver, stored in the gallbladder. And so we know sometimes issues with liver, issues with the gallbladder can arise depending on a person's diet and their cholesterol levels and so on. And so just that really important role that these bile salts can get in and separate, get between lipid particles, so that the lipid particles then will react with the solution that they're in. So, too much cholesterol in your bile, now you're going to get gallstones, right? Ideally, you can change things in one's diet rather than having to have a gallbladder removed. So, too much cholesterol, right? Which could be that there's just too much cholesterol, or it could be you're not processing efficiently. So if your concentration of bile salts is too low, then you're not getting enough separation and that cholesterol stays clumped together and makes those stones. Alright. So hopefully none of you have actually had this experience, but I think you're well aware that it certainly involves a lot of pain. These stones are forming and they're big, and so they're going to get lodged in the duct. They can't move through like they normally ought to. So you got a few things going on. One is, right, they're basically stuck. Another one is, because they're stuck in a passageway, the stuff that's supposed to get through the passageway now starts backing up. So a few things going on here. We know cholesterol also makes those steroid hormones. And if we look at our adrenocorticoid hormones, we said produced by the adrenal glands, and we separated those out into looking at sodium, chlorine, potassium, mineralocorticoids. So we know we need to not just stay hydrated, but we also need to have proper electrolyte levels, and this plays into that role with the mineralocorticoids. Glucocorticoids, cortisone, cortisol. So cortisol is increasing glucose and glycogen concentrations, which has, in the biochemistry, this is involved in anti-inflammatory effects, but that also means it's important just in our regulation of glucose and glycogen in the body, what our levels are, for all the various purposes that we use them for. We know glucose is our main energy source. Our sex hormones, so this just shows us the pictures of them, right? And we actually use progesterone in order to make testosterone, estradiol. So cholesterol to progesterone, and then we go on down the pathway. So if we were to draw just a map, we would start out with cholesterol, run a chemical reaction, and get progesterone. So that means that males and females both are producing progesterone, and then from there, females will use a certain amount of progesterone and then of course convert some of it into estradiol, and the males are going to be converting progesterone into some testosterone. And so it's actually all coming from that same pathway. We all have the stuff to make either one, so it actually becomes smaller differences than we think of typically in terms of males and females and the sex hormones and how much of what set you have, what your ratios are. Oh, and I was just going to say, I don't know if you remember that story from the Olympics of the lady from Africa. I can't remember how many years ago it was, and she naturally had very high levels of testosterone, and so she was doing really well in the Olympics, competing against women, and they were actually trying to make a decision about disqualifying her or not disqualifying her because they tested and they determined that she wasn't bulking up, she wasn't using any anabolic steroids, but she just naturally had a high level of testosterone, and therefore that made her faster. So again, just back to that point where we all have everything in different ratios, and so the tone of our voice and our hair and some of those other male versus female characteristics just come down to that difference of the ratios that we might have in our body. Anabolic steroids, of course, we're using synthetic chemicals to try and get some of those same reactions, teenagers wanting to bulk up, or a lot of our sports players wanting to bulk up. I'm sure you're likely well aware of the fact that they don't work the same way in the body, and the concentrations being taken don't match that which we would normally have, and a lot of those big downsides that this can cause, whether it's men developing breasts, getting higher voices, the balding process happening very quickly, and certainly there are a number of genetic illnesses, genetic diseases where some of the hormonal processes are not working right, or there might be some chromosomal issues where someone naturally has male pattern baldness but they're female, or some other things going on just because of the ratios of those hormones in the body and what the body's producing and what the body's breaking down. Okay, so if we switch to the prostaglandins, and we said they come from arachidonic acid, and we looked at the interesting shape. So it's cyclic in terms of the idea that it wraps around, right? But it's not actually cyclic in that it's not bonded at the other end. So we're starting with arachidonic acid, and we make these PGEs, but we'll notice they still have a methylene and a carboxylic acid end. We haven't actually done anything with that end. We are at the other half of the molecule, the other part of the molecule, creating a ring formation. So we're getting a ring down here to make it cyclic, and you'll notice these rings are slightly different from each other, which makes these chemicals slightly different from each other, but we're not doing anything at the other end of the chain, even though it wraps back on itself. So just at the one end. Alright, so we looked at some uses of the prostaglandins, and this is just a little bit more information on them. So PGE2 and PGF2, inducing labor. But then you can see PGE2 is also an aerosol form used to treat asthma. So we're seeing some molecules that can have some quite different effects in the body, depending upon which part of the body it's working on. So is it relaxing the muscles around the bronchial tubes? Are we looking at what's happening down in the pelvic area? So some really different functions even with the same molecule. Gastric secretions. We want to inhibit some of those gastric secretions if people are having issues with ulcers.